Hello students, today we will discuss G proteins and G protein linked receptors, their structure and general mechanism of action. First I will tell you about receptors. Receptors are a special class of proteins that function by binding a specific ligand molecule. Ligand molecule, if you remember, we have already discussed in our previous lecture, is a chemical messenger released by one cell, right, to signal either itself or a different cell. Just look at this diagram. This is signaling cell, right, that release ligand or chemical messenger, and second one is target cell that receive the chemical messenger. Ligands are released by this signaling cell and received by target cell, as target cell has receptor on its surface, and this ligand will bind to this receptor. When ligand binds to its receptor, the receptor can change conformation and transmitting a signal into the cell. In some cases, receptors will remain on the surface of the cell and the ligand will eventually diffuse away. In other cases, ligand binding to a receptor triggers a series of events leading to internalization of the receptor. Transmembrane receptor proteins are embedded in the phospholipid bilayer of the plasma membrane. Right? Now, types of receptor. There are two types of receptor. Internal receptors, also known as intracellular or cytoplasmic receptors, and the second type is cell surface receptors, also called transmembrane receptors. Internal receptors are found in the cytoplasm of the cell and respond to hydrophobic ligand molecules. Internal receptors have three core domains: a ligand binding domain, DNA binding domain, and an amino terminus. Amino terminus interacts with the gene transcription machinery. Once inside the cell, many of these molecules bind to the proteins that act as regulators of messenger RNA synthesis to mediate gene expression. Now, what is gene expression? Gene expression is a cellular process of transforming the information in a cell's DNA into a sequence of amino acids that ultimately forms a protein. Second, cell surface receptors. Cell surface receptors are cell surface or membrane anchored or integral proteins that bind to external ligand molecules. This spans the plasma membrane and perform signal transduction. Each cell surface receptor has three main components. First is an external ligand binding domain or extracellular domain. Second is hydrophobic membrane spanning region. And the third one is an intracellular domain inside the cell. There are three general categories of cell surface receptors. First is ion channel linked receptor. Second is G protein linked receptors. And the third is enzyme linked receptor. Today we will discuss G protein linked receptors only. Before starting with the G protein linked receptors, first I will tell you about what are G proteins. G proteins are specialized proteins with the ability to bind the nucleotides guanosine triphosphate, GTP, and guanosine diphosphate means GDP. Right? G protein is heterotrimeric. Just look at this diagram. This is G protein and is made up of three different subunits: alpha subunit, beta subunit, and gamma subunit. This is G protein. This is plasma membrane, and here is G protein coupled receptor, right? Now just look at this second diagram. This is G protein, and this form of G protein is inactivated, right? In its inactive form, GDP is bound to the alpha subunit of G protein, right? Once G protein binds to receptor, the resultant shape change activate the G protein, means releases GTP and picks up GTP. When GTP binds to alpha subunit, this form of G protein is called activated form, right? And this activated G protein then interacts with either an ion channel or an enzyme in the membrane. Now, G protein linked receptors. G protein coupled receptors are the largest and most diverse group of membrane receptors in eukaryotes that respond to a wide range of external signals. Now, this is G protein linked receptor. Look at this diagram. They are integral membrane proteins that contain an extracellular amino terminus, this part, 7 transmembrane alpha-helical domains. These are 7 transmembrane alpha-helical domains, 1, 2, 3, 4, 5, 6, and 7, and an intracellular carboxy terminus, this part, right? Actually, this G protein linked receptor is a single polypeptide chain that is folded into a globular shape and embedded in a cell's plasma membrane. G protein linked receptors work with the help of G protein. That's why G protein linked receptors. G protein linked receptors bind a ligand and activates a G protein. So G protein linked receptors just act like an inbox of messages in the form of light energy, peptides, lipids, sugars, and proteins. These messages inform cell about the presence or absence of life-sustaining light or nutrients in their environment, or they convey information sent by other cells. Now, structure of G protein linked receptors. As I already told you, G protein linked receptors contain an extracellular amino terminus, this part, 7 transmembrane alpha-helical domains, an intracellular carboxy terminus, and 3 intracellular loops and 3 extracellular loops, right? A transmembrane region crossing this lipid bilayer 7 times. You can count 7 times. Hence, they are also referred to as 7 transmembrane receptors, right? Now, G protein linked receptors contain extracellular and terminus domains, 3. These are ECL1, ECL2, and ECL3. These are extracellular domains, right? Of variable size, ranging from 154 residues to 36 residues, and an intracellular carboxy terminus domains, 3, means CL1, CL2, and CL3. These are intracellular domains, which are involved in several aspects of GPCR signaling. Now, extracellular loops, these are can be glycosylated and contain 2 highly conserved cysteine residues, which build disulfide bonds to stabilize receptor structure. Extracellular loops actually form part of the pockets at which signaling molecule bind to GPCR. Some GPCR also contain a cysteine residue and in the C-terminal domain, which serve as a site for palmitylation. This can create a 4th intracellular loop here because of the ability of palmitylation, palmitylated cysteine to insert in the plasma membrane. So C-terminus may be involved in interaction with other proteins that mediate GPCR signaling. Now, as GPCRs have no integral enzyme activity or ion channel, therefore all their downstream effects are mediated via their G proteins only, right? Now, plant GPCR and its role, right? Now, there are more than thousands of GPCRs known in humans and animals till to date. GPCRs in plants are not well characterized as compared to GPCRs from animals, right? Arabidopsis, our model plant, contains a single regulator of G protein signaling protein, that is RGS1, which is shown to stimulate the intrinsic guanosine triphosphate activity of G alpha. This RGS1 also contains heptahelical domain, RGS box domain, and might be functioning as a receptor or co-receptor. Now, till to date, there has been only one putative GPCR, means GCR1, identified and experimentally investigated in model plant, that is in Arabidopsis. Using a different bioinformatics tool, it was shown that there might be as many as 394 divergent GPCR candidates in Arabidopsis, right? Signal recognition of GPCRs is typically coupled by heterotrimeric G proteins to downstream effectors like animal GPCRs. Now, research studies indicated that GCR1, unlike typical 7-transmembrane receptor, as we have studied in animal, apparently acts independently to heterotrimeric G protein in at least some aspects of seed germination, suggesting that this alternative mode of 7-transmembrane receptor action also functions in the plant kingdom. Now, various plant responses that are affected upon genetic knockout of alpha, beta, and gamma subunits of G proteins suggested that there might be existence of more plant GPCRs which may be discovered in future, right? Now, a general mechanism of signal transduction through GPCR and G proteins. So actually, mechanism of signal transduction starts with ligands binding, right? So first, a ligand bind to the extracellular portion of G protein coupled receptor, binding either at N terminus or a binding site within the transmembrane region, right? When signaling molecule binds to G protein coupled receptor, the G protein alpha subunit exchange GTP for GDP, means binding at an extracellular ligand binding site actually causes a conformational change, right? Just look at this diagram here. Here the signaling molecule binding it receptor, right? It causes conformational change in the GPCR, resulting in the release of this GTP from the alpha subunit of G protein, right? So here G protein coupled receptor acts as guanine nucleotide exchange factor that promotes the exchange of guanosine diphosphate, means GDP, to guanosine triphosphate, means GTP, right? Now, this released GDP is then replaced with GTP, right? Now, these alpha subunits now interact with their relevant effector and cause downstream effectors, right? Downstream effects means response. Now, to prevent excess signaling, GPCR activity can be switched off. How? With the help of GTPase enzyme. GTPase enzyme catalyzes the breakdown of this GTP into alpha subunit, means on this GTP on alpha subunit into GDP and PI. This GDP increases the alpha subunit's affinity by this alpha subunit's affinity for beta gamma subunit, allowing the reformation of this heterotrimeric complex of G protein, right? And this G protein then reassociate with the transmembrane receptor and reforming GPCR for the next ligand binding, right? So first, signaling molecule binding, right? It causes conformational change. GDP from this alpha subunit then dissociate. First, this GDP is replaced by GTP, and then this alpha subunit dissociate from beta gamma subunit, and then it triggers a cellular response, right? Then this GTP is hydrolyzed to GDP and PI, right? And affinity of alpha subunits for beta gamma subunit increases, and it allows reformation again of heterotrimeric complex of G protein.